talk today is called Body Fluid Proteomics, Insights into Metabolic Health and Disease. And I'm a postdoc in the Deschamaux group here at the Center for Basic Metabolic Research. And we're a group that's really situated in this program one that's focused on metabolic dynamics and organ crosstalk, which is why particularly I'm focused, and the subsection of the group is focused on body fluid proteomics. And yeah, but we apply proteomics technology to various sample types, and today I'll focus a lot on body fluids. And we're really in this nice collaborative environment where we focus a lot on acquiring samples, processing samples, and looking at optimizing workflows in this really nice collaboration with PRI here at the University of Copenhagen that's led by Michael Wittbier. Yeah, so yeah, like I said, one of the big questions that we focus on is like what's the best way of studying proteomes, especially plasma proteomes. And recently, myself, Jeppe, another postdoc from our group, and Atul came up with this review comparing different technologies, both MS, affinity proteomics, and comparing the pros and cons. And I think the consensus, I won't go into the details here so I could focus on the science, but we're really utilizing the complementary nature of these different technologies and different workflows to try and understand metabolism. Yeah, so this is just some in-house data to highlight that when we look at different LCMS-based approaches as well as affinity approaches, we get this really really nice complementary nature of looking at unique biological insights across the proteome. And I mean, I feel like every day there's a new plasma workflow comparison, which is super nice and informative, which really gives us insight into which way to move forward and how to apply it to different sample types or different clinical questions. And today I'll focus my talk on mapping the secretome following exercise. So I gave a very broad title because we do a lot of different types of studies, but one of the key focuses of the lab is understanding exercise-induced secretion of different proteins. And to understand why we study this is we know, and it's very well established, that exercise is medicine, and that even slight improvements of moderate to increases in moderate to vigorous physical activity per week can reduce all-cause mortality, and more importantly, can also improve quality of life, health span, and reduce non-communicable diseases. And a major focus of our research, again, is to understand what this medicine is. And we study this through by trying to quantify, identify different exercise-induced factors, particularly proteins, and how they change with acute exercise across different body fluids like plasma, saliva, and urine. And we've been quite inspired by the work that has been done in the past, particularly this quite landmark paper by the Snyder Group, looking at the molecular choreography of acute exercise, which has highlighted a lot of different molecular features and how they respond to acute exercise. But we think that there is not a large focus or a low coverage of the proteome, particularly in plasma, and not a focus on other body fluids that might give bigger insights into what proteins might be important for the long-term health benefits of exercise. I think we're in a super nice position now with the method and technological advancements, as well as availability of data from different atlases and cohorts to help us, one, prepare the samples at scale, utilize different body fluids like urine, and then more importantly, understanding the health benefits of these factors that change with exercise. And I'll start off with talking about a preprint that we just put out a few months ago, answering this specific question. And in this initial discovery cohort that I'll talk about, which we did in collaboration with people at CFAS and Bente Carlin's lab, Peterson's lab, we took 25 healthy adult males in this initial discovery cohort and monitor and took plasma, saliva, and urine across 24 hours with urine only at pre, post, and 24 hours, and then plasma and saliva across 6 different time points. And on our workflow, we required in our LCMS approach only 1 microliter of plasma, but we also did affinity-based Olink proteomics on it, required 40 microliters. And then we also utilized 20 microliters for in-solution protocol that we used, and then 100 microliters for the urine for this HYLIC protocol that we used in urine. And in this study, we found that there's a robust yet distinct dynamics post-exercise across these different body fluids, namely plasma. When we measure it with affinity proteomics, we see almost 3,000 proteins changing immediately post-exercise, and over 3,000 changing across body fluids immediately post-exercise. And when we look particularly not just at time point specific effects, but when we cluster these proteome trajectories across this high granular monitoring that we did across the study, we can really resolve the differences between these body fluids and how they respond to acute exercise, where we see this robust and sustained change in saliva, whereas plasma, we have a very quick and dynamic change that basically comes back to baseline by 3 hours post-exercise. And today I'll focus just on a few of the highlights from this paper, just to keep it focused because I only have 15 to 20 minutes. But I think one of the most interesting findings from this work is in the saliva data, where we see this robust effect of almost 600 proteins changing, and we find some really interesting clusters of this dynamic proteome change, particularly in cluster 1, where we have this initial increase that's sustained to 24 hours. We see some interesting terms like peptide hormone processing, as well as sensory perception of taste. And we're quite fascinated about this because it's something that I think has been quite overlooked in the exercise field, and how appetite or satiety changes with acute exercise, and the link between the change in the saliva proteome. And when we do some different enrichment analysis, leveraging open source atlases like the Human Protein Atlas, we see that the proteins that are changing within this cluster 1 are highly linked to secretion from salivary glands and serous glandular cells, and that a majority of these proteins are known to be secreted, validating our approach as well, and some of these targets that we're quite interested in. Another fascinating thing about this cohort is the paired body fluid sampling, where we can look at how paired proteins change across saliva and plasma, or urine and plasma and urine saliva, to highlight this coordination across different body fluids. For example, when we look at one protein like MANC1, we see that there's basically no correlation at baseline or 24 hours post-exercise, but there's this coordinated control and association between the saliva and the plasma levels of this protein following exercise, which we think is quite fascinating, suggesting some either coordinated intertissue regulation of certain proteins. And I think another important thing, which I talked about before, is the goal of trying to link these changes to their importance in health and disease. And our group has published some work on salivary proteomic signatures of type 2 diabetes. And here, this enrichment analysis that we've been developing in collaboration with the Rasmussen group and Justus here is this disease enrichment analysis, where we take these protein signatures of these different temporal clusters and do an overrepresentation analysis on these different diseases. And what we find is that in this cluster 1 that's increased, it's highly linked to proteins that are also associated to incident disease in the UK Biobank Protein Disease Atlas, which we find quite interesting, suggesting that proteins that are changing with acute exercise are also linked to type 2 diabetes, suggesting some type of ability to modulate these proteomic signatures. Moving on to some other studies that we're doing is just to highlight the efforts in this area, is that we're also working on trying to validate that the origin of some of these proteins. So I didn't show you the plasma proteome data, but we want to try and find that validate that the proteins that we're linking to certain tissues in the plasma proteome are in fact coming from those tissues. So we're conducting some arteriovenous sampling. So, for example, we've conducted studies where individuals undergo an exhaustive exercise bout, and we'll sample arteriovenous plasma across a working muscle at baseline and then following exercise. And this work has been really led by a postdoc, Søren Jensen, and it's just to emphasize that we're able to disentangle which proteins are actually being released versus uptaken into working skeletal muscle post-exercise, which will help us get down to which proteins to follow up on, which ones are actively secreted during acute exercise. Another major effort of the group is to also try and map some of these proteins to a more specific secretion mechanism, which is through extracellular vesicles. And we think that this is a nice approach also to try and see which tissues are communicating or releasing certain proteins or different molecular features during acute exercise, that we can again get down to which ones are maybe actually mediating health benefits. And in these studies, we're really leveraging size exclusion based chromatography, and we've done a lot of method development on this approach as well. And in this study design, it's very similar to the discovery cohort. It's a sub cohort of actually that same cohort, where we're taking plasma across the acute exercise bout at 5 different time points and performing size exclusion chromatography to conduct proteomics, lipidomics, and small non-coding RNA sequencing on these samples. And in this workflow optimization, we wanted to try and see the balance between throughput and the purity of extracellular vesicles. So we've done a lot of work on different workflows, like leveraging maybe potentially this from Resin Bio, the SAX-based protocol, this magnet workflow. But what we find in this work is actually that the size exclusion based protocol and the SAX protocol are identifying distinct proteomes, and that the size exclusion has a relatively higher number of EV positive markers. So we think this is a more cleaner approach, and maybe it's not as high throughput, but maybe it help us study biology versus the clinical utility of leveraging the SAX protocol for high throughput sample processing. And for example, when we conduct these experiments, we also do this affinity column removal of the top 14 plasma proteins to compare the EV specific changes in the acute exercise proteome versus EV specific changes. So when we do the size exclusion chromatography, we take the initial 5 fractions, which is the EV enriched, compare also the later EV depleted fraction as well as the plasma fraction. What you can see is that we get this really nice high purity in this P1s. This is a TEM image here of these cup-shaped like vesicles. And again, just to validate, we see that there's a high proportion of EV specific markers in these P1 fraction, or the EV enriched fraction. And when we look at the multiome response to acute exercise, we see this quite different response depending on what type of omics we've characterized them by. So for example, the lipidome is quite robustly changed, whereas we have this early and later response in the transcriptome, whereas the proteome is quite robustly changed 3 hours post-exercise, which quite contrasts what we found in the plasma proteome in our earlier study in the body fluid proteomics study. Again, when we look at these multi-omic temporal trajectories, we see that when we integrate these omics, we find that there are protein-specific clusters, whereas as well as some clusters that have a coordinated response, like in cluster 5. And when we look at the overrepresentation analysis of these clusters, we see some quite, again, interesting terms, like in cluster 1, where we see an increase in terms like fatty acid metabolic processing. And what was fascinating, I think, about this identification of this term is that it also coincided with an increase at the same time point, 3 hours post-exercise, in triglycerides, peaking at 3 hours post-exercise. And what was really nice also to see, just in general, which is known in the field, that EVs are released, increased post-exercise, is that we see also an increase in ceramides, which are a known and key component of EV biogenesis and membrane structure. And when we do this EV enrichment tissue of origin analysis or tissue enrichment analysis, we see that cluster 1, that one that was increasing, is associated with skeletal muscle, proteins associated with high levels of expression in skeletal muscle, and that this was, when we do a cell type-based enrichment, specifically, these are associated with cardiomyocytes. Another key feature to all of these studies is try and to get down towards which exerkines or which proteins are actually mediating any effect. So in totality, we've identified 3,500 or so proteins that are changing with acute exercise, but it's really hard to get down to which ones you want to follow up on. And some of the work that we're now trying to do is trying to integrate known atlases, and we've been collaborating a lot with the Loose group to do this, where we're combining co-localization analysis from this Open Targets platform, where we're co-localizing these cis molecular traits with known traits in open targets. So, for example, like different diseases or physical activity parameters, to try and get down to which proteins might be mediating some of these health benefits of acute exercise. So the general hypothesis here is that if you have a genetic variant that increases the expression of a certain target, like a certain protein, and it's also having a same shared signal with a trait of interest, like diabetes, for example, maybe this is mediating some of those effects with acute exercise. And we've done some of this analysis to integrate our known proteins that are changing with acute exercise and this colloq data. And what we find is that a lot of the traits and a lot of the proteins that change with acute exercise are forming a network that's really revolving around metabolic traits. But then there's some other sub-communities that are, or modules that are forming that are related to physical activity or muscle function, cardiovascular disease, neurological body composition, or endocrine and digestive diseases. And when we look deeper into these networks, we can form sub networks that are specific for certain traits, like metabolic traits, and filter for those that are only that have a known drug target. So we have a starting point on which proteins might be interesting to follow up on as a viable already repurposing for target validation and influencing certain traits that we're interested in, like metabolic traits. So some key highlights from this work is that body fluid proteomics can uncover distinct temporal trajectories. Tissue enrichment can uncover contributors to exercise-induced extracellular vesicles. And I think co-localization or genetic integration in general can identify druggable exerkines or exercise-induced proteins that may regulate health and disease. And just to mention that this is a super collaborative project spanning many groups and a lot of people that made this happen. So I want to thank all of them. So CFAS for a lot of the sample collection, the Deschamuk group in general, and particularly Jeppe for being a lead author on the initial paper as well. Same with Naya, and then the Rasmussen group and the Luce group for being real key collaborators on the bioinformatics analysis. Yeah, so